Our next presentation is Ray Therapeutics, and I'd like to welcome Peter Francis, Chief Scientific Officer and Medical Officer, to the stage.
Thank you very much for the opportunity to speak this year. It's a pleasure to be here in a rather hot Phoenix. So Ray Therapeutics, actually since last year, is now a clinical stage biotechnology company based in San Francisco and Berkeley, and then we have a satellite in Irvine, California. We have a mission to restore vision to patients with retinal degenerations. We have two lead assets, RTX-015 for retinitis pigmentosa and other inherited retinal degenerations, and then RTX-021 for those patients with failing central vision due to macular disease. Our big differentiator in this space of optogenetics or visual optogenetics is our highly bioengineered optogenetic medicine, which has been optimized for human vision. We're using also advanced AAV vector technology to allow simplified intravitreal delivery to the eye. And we have the realistic prospect of not only restoring light sensitivity to blind patients, but also the potential for higher visual function as well. We've raised more than $100 million thus far, which will support our programs through late-stage development, including non-dilutive funding from the California Institute for Regenerative Medicine, CIRM. We are currently in a run-in study for our RTX-015 lead asset, and the interventional study is enrolling as we speak. This is just a summary of where we are. Our RTX-021 second program is in late IND-enabling studies right now. We hope that that will enter in clinic in the first half of 2025. We also have a discovery program in the neurological space that we're very excited about. A few words about optogenetics. It's pretty much a revolutionary technology. It's been around, it's beginning to mature now. It's been around for over 20 years. It's based upon the identification of unique transmembrane proteins that are found in nature, unique by virtue of the fact that they are channel proteins but gated by light, so they can be switched on and off using incoming light. This has been harnessed as a technology for vision in blind patients who have lost a significant number of their light-sensing cells, their photoreceptors in their retina, but where a substantial number of other neural cells, which continue to be wired up to the brain, exist, and we can make those surviving cells light-sensitive. We've also taken, we think, a pioneering step, which we recognize that measuring endpoints, clinical endpoints, in patients with very low vision is challenging. While we have some excellent endpoints to look at, for example, visual acuity and visual mobility tasks, many of these patients need a further refinement of these endpoints in order to be actually able to do them and have something measurable. So we opened up our rather grandly named Vision Research and Assessment Institute in Irvine, California, for patients to come to a comfortable, familiar environment. It's staffed by low vision experts, and we have refined those endpoints so they're now more suitable and can measure the outcomes better in patients that are the target group for our therapies. We now have IRB approval to enroll patients with multiple indications, retinitis pigmentosa, Stargardt's disease, macular degeneration, and so on. And this has been this program has proven to be a tremendous success thus far. So a few words without me speaking on our lead asset. Optogenetics is a novel technology utilizing light-sensitive, naturally evolved proteins, channel rhodopsins, to control neuronal function with light. Visual optogenetics can potentially restore vision in people with retinal disease by converting surviving neurons into photosensing cells. Most treatments aim to slow vision loss. Optogenetics can restore vision, is not dependent on any gene or mutation, and has the potential to work on all forms of retinal disease, including retinitis pigmentosa, Stargardt disease, and the geographic atrophy form of age-related macular degeneration. Ray Therapeutics has bioengineered the next-generation channelrhodopsin-based gene therapy treatment for human vision, RTX-015. It is administered through a safe, quick, minimally invasive intravitreal injection in the doctor's office and repurposes the retinal neurons that survive in retinal diseases into light-sensitive photoreceptors. The retinal ganglion cells then directly relay visual signals to the brain for interpretation as vision. RTX-015, a major advancement in the field of optogenetics with the potential to dramatically improve the quality of life for those affected by retinal degenerative disease. And so now a few words about our second product, newly released data on RTX-021, which is a low-dose intravitreal gene therapy for the treatment of those patients with macular and central visual function, such as age-related macular degeneration, the dry form of that disease, which is in fact the leading cause of irreversible blindness in patients over the age of 60. It's estimated that there are more than 1 million patients in the United States alone that suffer with this condition and would be suitable for optogenetic therapy potentially. There is a juvenile form of macular degeneration called Stargardt macular dystrophy, which is a monogenic disease caused by mutations in the ABCA4 gene. Its prevalence, of course, it is a rare disease, is much lower, fortunately, for the children that are involved, but it still numbers a total of about 40,000 patients, and we estimate that up to 50% of patients with that disease would be suitable for optogenetic therapy. There are no treatments for Stargardt macular disease that have been approved. There are for geographic atrophy, where it's exciting last year with the approval of 2 treatments. But these treatments slow down the rate of further geographic atrophy development. So our therapeutic approach would be potentially adjunctive in potentially improving vision in those patients while they might receive these treatments to slow down the general progression of the disease. As I mentioned, it's an intravitreal injection. This shows a stylized diagram. We've worked hard over the years to develop a highly specific, cell-specific, but highly expressing promoter for on-bipolar cells, which are a specific retinal subtype. And so we can achieve high levels of selective expression in these on-bipolar cells, which we know exist late into the degenerative process in conditions like geographic atrophy and Stargardt macular dystrophy. As I mentioned, it's given as an intravitreal injection. The gene therapy is designed, the capsid we're using achieves very high levels of transduction in the inner retina, which is the target here, and expresses our optogenetic protein, which is then activated by incoming light. So what of our data? This is data actually in an animal model. This is not some sort of in vitro measurement of light sensitivity. This is actually asking the question, how well do the animals see? And you can see here on this simple diagram that the animals, the threshold at which the animals are able to detect light using a very standardized objective testing paradigm is very low. It's down towards very much sort of, I guess this lighting level here would be the equivalent, or sort of street lighting at night. So it's not active right down to total darkness, which we actually think is an advantage, but it is active. And then it retains a very, very high dynamic range right up to bright lighting. So essentially, this optogenetic protein should be active throughout all ambient lighting without the need for any type of light-enhancing eyewear, which has been needed for optogenetic therapy to work in the past. And then when we look at higher visual functions beyond just light sensitivity, now looking at visual acuity and contrast sensitivity, we can achieve more than 50% of the normal visual acuity of these animals. And these are animals that were previously completely blind, so they have a very limited capacity for visual improvement. And then when we look at contrast sensitivity, we can create almost normal levels of contrast sensitivity, which have not been seen with other optogenetic approaches. So we're very excited by that prospect. This is exactly the kind of vision that patients with macular degeneration would benefit from. So here's our value proposition. We have a high-resolution optogenetic protein delivered as a single low-dose intravitreal gene therapeutic, specifically on bipolar cells. We're looking for a visual improvement, which actually makes the clinical development pathway very attractive because it's a near-term visual improvement, which then should show durability. It would be in clinically approvable endpoints, and so it streamlines your clinical development program substantially. We believe we've already conquered the CMC aspects of this in terms of high levels of productivity using an already scaled commercial process with our lead partner in this process, which is Forge Biologics. And as a result, we're very excited by the potential for RTX-021 to restore vision in patients with a potentially large market indication. And with that, I'll say thank you so much. And never to be forgotten are the patients who are now contributing substantially to our work. And we thank them and their families and caregivers for all the time they spend with us and their commitment to our programs. Thank you very much indeed. And I'm happy to take any questions. We've got a couple of minutes. For 015, which is targeting retinal ganglion cells, they do persist right up until the end stage of disease in conditions selectively like retinitis pigmentosa. And our enrollment criteria, we've worked with a reading center to define the ability to measure your retinal ganglion cells on what's called an optical coherence tomography test, which is a non-invasive, clinically based test. And so all the patients that get enrolled, we have a measurable and quantifiable level of retinal ganglion cells in them. For the 021 program, absolutely the ganglion cells are preserved at least until very much the end stage of the disease in those conditions. And we also know that the bipolar cells are present for the 021 program as well. That's not so easy to demonstrate on an OCT. But we're working on a technique called adaptive optics OCT, which gives us higher resolution, and so we should be able to define those patients very clearly for the clinical program. Thank you. Any other questions? And with that, I'll say thank you so much again.